I'm a basic scientist. My lab is at the Duke Molecular Physiology Institute. But more importantly, I'm the Director of Graduate Studies for the University Program in Genetics and Genomics. So when, this is obviously not way out of control, it's a lot larger than we first envisaged. So this all started Monday last week, and so the program, the UPGG, was contacted by the Wall Street Journal, and the journal was interested in a paper that just dropped in the Associated Press, and they're asking for one of our faculty to comment on this paper. And so on Monday, Liz and Amanda, who are the administrators of the program, mailed me and said, is there anyone in the UPG faculty who could speak to it? And obviously you'll find out why we sent it to Charlie in a minute when he talks. So that was Monday. Come Thursday last week, so a week ago, I mailed Charlie and said, you know, did the Wall Street Journal ever contact you? And he said, yeah, I'll tell you about it when I get back from Hong Kong. I suddenly thought, ooh. I said, are you at that meeting? And he said, yes, I am. And I immediately thought it would be awesome to have someone who was in the room when it happens, hashtag Hamilton. So I talked to Charlie, and the first discussions were, could you speak to a group of UPG students? Because today is all about the students. So faculty who are hoping to get a question in, I'm sorry. It's just the way it goes. And he said, yeah, love to. And so I reached out to David Tobin, who's the DGS of MGM. And David said, yeah, it'd be great if the MGM students could come in as well. So Charlie at that point thought it was just a fairly small room with two programs of students that he would be talking to. And then, where's Crystal? Crystal, at a meeting, suggested that perhaps we should, you know, it'd be good to have someone like Ginny Fuda on the panel. and suddenly we thought, hey, why not have a panel? And so we have a panel host. Is Vandana Shashi? Where is she? Is she here? Not yet. All right. If you know Vandana and she turns up, we have a seat at the, well, obviously not that seat just yet. We got a seat at the front for her. And so we had a panel, and I cast around, and the names of the folks who are on the panel are up on the table here. And suddenly it was sort of word of mouth, and graduate students and students are much more adept at spreading the words than the PIs are. And suddenly it went beyond just UPGG and MGM and spread much more widely. And then Misha had the great idea of getting the Duke Initiative and Science and Society guys on board, and they were advertising, and suddenly we got a buttload of people in this fairly large room. So that's how it happened. I've been in this room with a similar sized crowd. When I remember, it was Sidney Brenner who was talking. And in that instance, we have, the investigator with decades-old track record of science, of publication, of known work. But here, we're talking about a publication that's not even in bioarchives. We've seen data that's presented at a talk that we're going to discuss later on. And it's from a fairly unknown individual in the field. So we've gone from our usual distinguished lecture series of talking about science that we know and understand. Here we're talking about science we don't know much of, and we're trying to think and get our heads around the implication of what we're doing. So I thought it'd be great if Charlie could give a talk first. He's a phenomenal science in the program. He's a scientist and faculty member of BME. So he's going to frame the sort of discussion for us that we'll have later on. But first I'd like to acknowledge the guys who actually got this all ready in a week. Okay? So Amanda and Liz, who organised stuff and kept me calm as this kept on snowballing. Christian Thol from MGM. David Tobin, of course. So together we worked out what the panel should contain. Crystal and Imam for the great suggestions. You guys have been posting us questions as well, so I have a whole list of questions, and we'll try to get to yours as well as you think of them as the guys are talking. Also, Ben Shepherd and William Rogers from the Science and Society Initiative. This is actually being broadcast live via Facebook, and it'll be recorded. And of course, Charlie for actually agreeing to this in the first place. It should have been about 1/10 of the size that it was. He said, I had tickets to the basketball last night, and I very meekly called him up to see if he wanted one. But he was giving another talk, so I still owe him a favour. And of course the panel. So when David and I reached out to the guys who are going to be on the panel, there was absolutely no hesitation about joining the panel. So we've got, we'll get the guys to introduce themselves after Charlie's talk, but I hope you'll see, if you haven't already looked at the links that we have on the page, that they really cover the gamut of what this question is, this really, really important question is about. It's supposed to be a discussion, certainly talking to my 15-year-old in my family, talking to my lab group, talking to the guys at the DMPI. Passions are running fairly high with this, but we'd like to keep it instructive, objective, and informative, and again, it's just for the students. Faculty can learn from the students. We'll get the students to interpret for them later on. Okay, so with that, I'll hand over to Charlie. Good luck.
Thanks for the introduction, Simon. And you got the story mostly right, but when he reached out to me and said, would you be willing to get a cup of coffee with a couple of UPGG students, I said I'd be happy to, but I usually like to keep a low profile around controversial issues. but a coffee with a couple of students seems fine, so here we are. So I thought of this famous proverb, I guess, may you live in interesting times. I was trying to figure out what I was going to talk about, because I guess the idea here is it sounds like a blessing, but it's actually meant as it's referred to as an ancient curse, because interesting times usually refer to times of war and turmoil. So I thought it might be appropriate here. Also, given that the conversation may venture into conflicts of interest, I thought I should probably disclose a few things. So as Simon mentioned, we work in genome editing and other genome engineering technologies in my lab next door, and I do have financial interests in companies engaged in that area, and I'm involved in patents and patent applications involved in that area, but none of my work involves any research or other types of activities that has anything to do with human germline editing or human embryos. So what I thought, I'm going to try and get through these slides quickly so that we have time for the panel and Q&A, but I did actually think it was really important to take this time, mostly because probably many of you have read about the story, but I did want to put a lot of this in context because there's a lot going on in gene therapy, gene editing, somatic gene editing, germline gene editing, and I think that it's not always clear unless you're in the middle of it what all of the differences are and what the status of the field is. And what I, as someone working in the area, find critically important is to be able to make the distinction between some of these different things, both technically and scientifically, and also ethically. So I'm going to try and give a quick background on kind of where we are with gene therapy and gene editing. I'll talk about the rationale for the CCR5 gene as a target for gene editing for HIV therapy. And then I'll talk a little bit about where the field is with somatic cell gene editing as an introduction into what's going on and what has happened in this area of human germline editing. Give a little bit of summary of what's gone on at each of these two summits on human gene editing, and then quickly just describe this recent report of human germline editing and the concerns before we lead into the panel. In the context of gene therapy, I mean, I just want to make the point that gene therapy is an idea that's been around for a really long time. And has had a lot of ups and downs over the last 50 years or so. So this is an article in Science back in 1972 that was kind of proposing some of the first examples of gene therapy with the general concept that you could use good DNA to replace with defective DNA. So this is kind of the conventional idea of genetic modification of human cells for therapy, where there's a genetic mutation and you want to add an extra copy of that gene as a replacement to act as a therapeutic approach. And I think that something that even many people in the field of genetics and genomics don't appreciate is how much activity there has been in human gene therapy clinical trials. So we had the first gene therapy clinical trial for humans starting in the late 80s. And more recently, we've had more than 100, and especially recently, more than 200 different human gene therapy clinical trials start every year. So lots of activity in this space, and it's only growing as the field has kind of overcome many of the challenges and started to make a lot of really fantastic progress. And that progress has been not just clinical trials, but actually we now, in the last year or two, have the very first examples of approved products for human gene therapies. And that started with nucleic acid-based drugs for treating some horrible genetic human diseases, followed by genetically modified cells for things like cancer applications that have seen tremendous successes in the clinic. And then more recently, the kind of conventional idea of gene therapy that is gene replacement, where you're transferring an extra copy of a gene to a cell, in this case using a virus to deliver a gene to the retina that treats a rare form of congenital blindness. And then most recently, an example of, the first example of a small interfering RNA to treat another rare genetic disease. So now the point of all of that is that we've got a lot of success with conducting many different human gene therapy clinical trials, and now we have a new wave of products that are getting approved to take advantage of this technology and treat some of these diseases. All of these examples, though, either rely on adding an extra copy of a gene to cells to replace a defective gene, or relies on getting rid of a faulty gene product that's causing some type of problem. And that's all in contrast to gene editing, where the idea is rather than adding something, an extra copy of a gene, or rather than trying to remove a faulty gene, it's actually going into very specific base pairs, into 3 billion base pairs of the human genome, and exchanging, replacing, or removing very specific base pairs in a very specific gene at a specific site. And so that's generally what distinguishes gene editing from gene therapy. And it's important to appreciate how difficult this is as a technical idea, given that every cell's got 3 billion base pairs of DNA, and the ability to design molecular machinery to go in and find one of those base pairs, distinguish all the other A's, T's, C's, and G's from the genome, and change just that one is a very difficult task. And the way that that's been accomplished is by taking advantage of DNA repair machinery. So in the mid-90s, Maria Jason's lab showed for the first time that if you create a break in the DNA in a very targeted place, then you can take advantage of the natural DNA repair machinery to change the DNA sequence at that site. And I probably won't go through the details of the DNA repair machinery involved for the sake of getting to the panel discussion. But what will be most important for this presentation is this middle one here, whereas the kind of first applications of gene editing are occurring. So in this case, when you make a break in the DNA with, for example, an enzyme called a nuclease, then typically the cell will grab those two loose ends in the DNA and ligate them back together. And this happens all the time. We get breaks in our DNA every time our DNA is copied, every time cells divide, if you go out in the sun. And cells are very good at ligating these loose ends of DNA back together. However, if you're forcing the overexpression of an enzyme that's going to create that same break over and over and over and over again, eventually that cell is going to make mistakes. And those mistakes look like the addition or deletion, the insertion or deletion of one or more base pairs of DNA that we call indels, so little scars within that coding region. And so those indels, if you add or remove a few base pairs from the middle of the gene, you can potentially knock out that gene. And this is kind of the most common way that gene editing is being done. There's a variety of other approaches where you can make two breaks to remove segments of DNA, or you can take advantage of a process called homologous recombination to add or exchange sequences of DNA. But all of this gene editing machinery or concept relies on the ability to have this nuclease enzyme that creates a break at a very specific site in the genome. And I think that in some cases there's kind of this faulty idea that gene editing is something that's very new, that's only recently emerged in the last couple of years. And in fact, that's not the case, because as I'm showing here, Maria Jason's lab showed these studies in mammalian cells in 1994. But she did this in a system where it was kind of an artificial system that had been engineered to be able to create these breaks. What was needed to take advantage of this discovery was the ability to make enzymes that can cut anywhere in the genome at will. And so that kind of inspired a series of efforts to make new enzymes that can cut DNA at a very specific site. And these earlier versions of this technology that started to emerge in the mid-90s were things like meganucleases and zinc finger nucleases, more recently tail nucleases, or tailins for short. But all of those technologies, while they've been around for decades now, and there's a few people in the world that are very good at using some of these technologies, they're also very difficult to work with. So it wasn't really widely adopted as a technology for modifying DNA. And that all changed with this recent emergence of the CRISPR-Cas9 system, where you no longer needed to engineer new proteins to target different sites in the genome, but rather just had to change the sequence of a single short RNA in order to target this nuclease enzyme Cas9 to any particular site. So not only was it easy, cheap, fast, but it was also very robust. So as opposed to some of the earlier technologies where you had to screen through dozens or hundreds or thousands of different versions of those proteins to find ones that worked really well, with CRISPR, most of what you make actually works pretty well if you know how to use the techniques. And so for that reason, probably many of you and many of others have started to use the CRISPR technology to do gene editing for research and other applications. And so that's really what has generated a lot of interest in this field and gotten the technology into the hands of a lot of different people. So if you can change the sequence of the human genome in any way that you want, one of the most exciting things to do is to treat human disease. And that can be either correcting mutations that cause inherited genetic disease, or modifying the genome in other ways that can do things like prevent viral infection. And that's going to be the focus of much of the rest of the presentation. But this list here is just a sample of some of the many areas where there's both preclinical and now human clinical work in using some of the earlier gene editing tools that I described to treat human disease. So this example of preventing HIV infection is based on the discovery of CCR5 as the cell surface receptor, the protein on the surface of our white blood cells that is the primary molecule that HIV uses to enter into and infect these cells. And so that's shown over here in this schematic. You have the virus binding on the surface of the cells and using that to get into these white blood cells and establish an HIV infection. Now, one of the ways that actually CCR5 was discovered was a fascinating discovery that there is about 1% to 2% of the Northern European population, or those of Northern European descent, that naturally carry a mutation to the CCR5 gene, in this case to both copies of the CCR5 gene, that's known as the delta 32 mutation. So this is a mutation, it's a 32 nucleotide deletion in the CCR5 gene that then knocks out that gene. And this was discovered because there were some individuals during the HIV epidemic that were routinely exposed to HIV but did not establish an HIV infection. And the scientists and doctors studying these patients realized that they did not make CCR5. Now what was fascinating was because these patients were naturally null for CCR5, HIV could not enter into their T cells, but otherwise they were, to my understanding, pretty healthy individuals, which indicated that CCR5 as a protein, at least in adults, was extraneous to normal, healthy lifestyles, and that you didn't actually need this protein in order to have a healthy immune system. So that indicated that CCR5 might be a very useful drug target to prevent HIV infection. Now, before we get to how you might use CCR5 as a drug target, the idea of getting rid of CCR5 as a way to prevent HIV infection was kind of made famous or established in this very famous experiment where there was an individual in Berlin who was originally called the Berlin patient, but his name is Timothy Ray Brown. He was an HIV positive individual who then developed a blood cancer that was independent of the HIV infection. And the treatment for the blood cancer was to get a bone marrow transplantation. And the doctor who was doing the bone marrow transplantation had the clever idea of saying, well, if I'm going to switch out this person's white blood cells, why don't I find an individual who is one of these few 2% of the Northern European population that is naturally null for CCR5 and take their bone marrow and transplant it into this HIV positive individual and see if perhaps those cells will now be resistant to letting this HIV infection propagate. And in fact, that experiment was successful, and the work was done, I think, around 2008, and to my knowledge now, 10 years later, Timothy Ray Brown is still no signs of HIV infection. So that supported the idea that if you had an HIV positive individual and you could remove or swap out, remove the CCR5 gene or swap out cells that were null for CCR5, that you could potentially take away all of the ability of HIV to propagate in that individual and potentially treat the HIV infection. And so this is the New England Journal paper that came out in 2009 reporting long-term control of HIV infection following the bone marrow or stem cell transplantation with cells that were naturally null for CCR5. And so this inspired the idea among some individuals that you might be able to use gene editing for this type of application, because even though this worked to treat the HIV infection in this one individual, bone marrow transplantation has a lot of risk associated with it. In fact, I believe that Timothy Ray Brown later had graft versus host disease and other complications. So you can't just have a bone marrow transplantation for every HIV positive individual because of the concerns of things like graft versus host disease and taking somebody else's cells and putting them into your body. But it inspired the idea that maybe you could do this alternatively as an autologous cell therapy. So if you had an HIV positive individual, you could take their either their T cells that are the primary source of HIV infection, or even the blood stem cells, isolate them from the patient, use gene editing with any of the technologies that I just described a few slides ago to knock out CCR5, and now take those autologous patient-derived cells that are missing the CCR5 gene and re-administer them back into the patient. And so that was the idea, and it was originally shown that this works using human blood stem cells, but using mouse models, as a collaboration between Carl June's lab at the University of Pennsylvania and Sangamo Biosciences, which is the company that originally developed zinc finger nucleases as a human gene therapy. And so this was a study in 2008 showing that this worked in preclinical mouse models, and then that led to a series of clinical trials where they then pursued this in humans. And this is the first published results from those trials, again led by Carl June in collaboration with Sangamo Biosciences, and this work was originally published in 2014. So this was the first example, to my knowledge, of a gene editing clinical trial in humans. Of course, it was an ex vivo trial where they took the cells out of the body. It was a somatic cell gene editing trial where they were using cells of the body, not the germ cells. And that was the first of many trials, and I've given, this is not a comprehensive list, but another thing that I wanted to make clear is that there's a number of gene editing clinical trials going on right now for a number of different indications, but you might notice that many of the first wave of clinical trials were using this CCR5 as a target for this type of ex vivo somatic cell therapy, although now that's moved into a variety of other applications. And that includes both this type of ex vivo modification of T cells or hematopoietic stem cells, but also now in vivo gene editing, where viral vectors are being loaded with gene editing tools and administered into the body to go to places like the liver or the retina to treat different forms of genetic disease. So with that as a background of somatic, again, I want to emphasize all of that is somatic cell gene editing, modifying cells of the body in an individual that is affected with a disease that you do not expect to modify the germline, and thus you do not expect these genetic modifications to be passed on to the progeny of any of these treated individuals. So all of that is in contrast to really what we're here to talk about today, which is germline editing or embryo editing. So this schematic from this website helps to give a summary of how germline editing works. And the way that works is really by administering the genome editing tools to one-cell embryos following in vitro fertilization. So egg cells are fertilized in vitro, or IVF, in vitro fertilization, and then your gene editing tool of choice, including CRISPR-Cas9, is then injected into that one-cell embryo. And then at some point following that, the CRISPR system, or whichever gene editing tool you're using, may go in and modify the genes as you intend. But at the same time, that one-cell embryo is going to start dividing into 2 cells, 4 cells, 8 cells, et cetera, and eventually into a blastocyst that would be suitable for implantation and then taken to term for birth. And at some point after the 8-cell stage, it's possible to take one of the cells from the embryo and use that for DNA sequencing that would allow you to genotype what happened in that cell. And this is also how pre-implantation genetic diagnosis, or PGD, works. So if you know that you're a carrier for a genetic mutation, then, and you know that, for example, if one of your alleles carries the mutation and the other does not, you might use pre-implantation genetic diagnosis to screen out embryos that carry the mutation and only implant the embryos that do not have that mutation. But another important thing that this schematic confers is that you can sequence this cell, and the other 7 cells, or however other cells are left, will go and will develop into a normal, healthy embryo. But the sequence of this cell does not necessarily tell you what the sequence is of the other cells that are left behind in that embryo. And we'll come back to that in a second. So as the CRISPR system, as I mentioned, really got gene editing tools and technology into the hands of many individuals, the amount of work that was going on in the gene editing field really exploded. And many labs that never knew about gene editing then began to work on this. And one of the things that people were using was using this type of approach to make genetically modified animals to help for disease modeling and for basic research. So and in many instances, this type of technology has been used to make genetically modified mice and rats and other types of model organisms for research. But a major step forward was in 2014, when that same type of germline editing was used to make gene-modified cyno monkeys. So this was the first example of kind of gene editing in germline in a non-human primate. And so this, again, I think in these next couple of slides, some of the dates are important, or at least interesting. But this kind of set off the idea that if you can do this to make genetically modified non-human primates, then could this potentially also be used for genetic modification of human embryos? And since in vitro fertilization is so well established, and on the schematic I show you that basically what we're talking about in germline editing is simply in vitro fertilization plus an extra gene editing step, it became clear that this might be possible. And the community knew this, and I think that there began to be whispers that there were people out there trying to do germline editing in human embryos, and that led to editorials like this one in Nature, actually led by scientists at Sangamo Therapeutics, the same company that I mentioned was running clinical trials for somatic cell gene editing, really imploring the community to not pursue human germline editing. So this was published the following year on March 26th of 2015, and it turns out that they were right to be concerned because 5 days later, this paper was published in a journal called Protein and Cell, where a group in China had published the first example of using CRISPR to do gene editing in human single-cell embryos. So this was a concern that the community had, and this led to the following month editorial by a number of people in the field kind of commenting on whether human germline editing should be pursued. I should take a second to point out that this study, these embryos were never taken to term, and they intentionally used tripronuclear zygotes that are incapable of going to term because that was never the intent of the experiment. So they basically were interested in demonstrating human embryo editing, but not with ever implanting these embryos or taking them to term. But it was clear that that was a possibility. So, so the community kind of spoke out about some of its thoughts about where human germline editing stood at the time, and that led to later that year the first international summit on human genome editing. That was a joint effort between a number of the different national academies across the world, and in both US, China, and the UK, to talk about what should and shouldn't be done with human gene editing. And so this was the statement that came out of that meeting. And they talked about a couple of different ways. They basically discriminated the different ways that human germline embryo editing might be used or might be performed. And they wanted to distinguish these. And they started with talking about basic and preclinical research. And coming out of that meeting, their recommendation was that basic and preclinical research is clearly needed and should proceed. So that was an interesting conclusion, but it basically gave a green light for human embryo editing under the same types of regulations and stipulations that other human embryo editing might happen, namely that these embryos should be terminated before 14 days post-fertilization. So that was a step forward, or at least a step in one direction for the field. Then they also discriminated the clinical use for somatic cell editing, which is the clinical trials that I already described, where they're using the adult tissue-specific cells of an individual affected with a disorder. And they basically said that because the clinical uses are intended to affect only the individual who receives them, regulators can weigh the risks and potential benefits in improving clinical trials and therapies. So basically a green light with no ethical concerns for somatic cell gene editing. But then on the topic of clinical use, so therapeutic use of germline editing, they had a lot more to say. And to summarize just on this point, they concluded it would be irresponsible to proceed with any clinical use of germline editing unless and until the relative safety and efficacy issues have been resolved based on appropriate understanding and balancing of risks, potential benefits and alternatives, and importantly, broad societal consensus about the appropriateness of the proposed application, and anything should proceed under regulatory oversight. And at present, none of these criteria have been met for any proposed clinical use. So basically recommending that this should not happen, and then they also recommended the need for an ongoing forum. Now, interestingly, just to give you some context of how the field then proceeded in this area, as I mentioned, their recommendations gave a green light for human embryo editing for research use. And in fact, even though there was kind of a considerable amount of controversy when that first paper came out in Protein and Cell in 2015, in 2017, there were two more papers published in Nature, one from a group at Oregon Health Sciences in the United States and another group in the UK, in both cases doing human embryo editing for basic research applications and not taking these embryos past 14 days. So then 3 years later, they decided that it was time for the second International Summit on Human Genome Editing, and this was the meeting that happened last week. And I can tell you, as an attendee of the meeting and talking to other attendees, I think everyone went into this meeting expecting a fairly boring meeting. There's a lot of genome editing meetings, and a lot of them talk about the same things. And so a lot of us were looking forward to dim sum, noodles. By the time we got off the plane, these two articles had broken. Our emails were full of journalist requests and other things. So while we were on the plane, this first article from MIT Technology Review came out that basically an exclusive article that they had discovered that Chinese scientists are creating CRISPR babies. Interestingly, this article just inferred that someone might be working on this, or is working on this in China, but did not reveal that actually 2 babies had been born. But hours later, the Associated Press came out with their stories that a Chinese researcher claims that the first gene-edited babies had been born. So this obviously changed the whole context of the meeting that then started on the following day and generated a considerable amount of excitement and attention. I'm going to include, this is my iPhone video, just to give some context to it. I've been showing this to other people. This is the panel right before the talk that revealed the story about genome-edited babies. And if I can show this, half of the auditorium had been blocked for the press. And this is not like something I've ever seen before. So we've got the scientists are taking pictures of the people taking pictures that are lined up all up and down the auditorium. So I would go to the meeting during the day, and then since it was Hong Kong, I'd wake up very early the next morning. And so I'd go to the gym in the hotel to get some exercise, which had 5 or 6 different TVs in it, and all of them were showing the meeting from the previous day on the local news, which was a very different type of thing that I'm used to. So as many of you already know, the talk that came next was the reveal that this professor in China claims to have created the first 2 gene-edited babies. And before getting into some of the issues, I just want to come back to the schematic that I showed earlier that describes what germline editing, what the protocol would look like, and ask the question of, well, what could possibly go wrong? And it turns out that there's a number of different areas where this could go wrong. And almost all of these have come to fruition in the results that were described that I won't take the time to get into so we can get to the panel. But one issue is that in some cases you don't know the embryo genome sequence, not only because genome sequence might be private to that individual, but as soon as a cell starts dividing, the genome sequence actually starts to change. So that's one concern, although I do believe that they claim that they have sequenced the embryo, the individual embryo, the genomes of the parents ahead of time. I think that's true, but obviously you don't know which combinations of genes ended up in this one-cell embryo. So another problem is off-target effects. So you design your CRISPR system to cut at one site in the genome and change the DNA there, but you really can't fully at this point understand with complete certainty where else it might be cutting and changing the underlying DNA sequence. And so now one thing that you could possibly do is remove one cell and do whole genome sequencing of that single cell, which I believe is what they have claimed to have done. However, whole genome sequencing at the single cell level is a far from perfect technology. You get some information, but it's, you don't get complete information. And as I mentioned earlier, the genome sequence of this cell doesn't represent the genome sequence of all of these other cells necessarily. And that gets to the issue of mosaicism. So if you inject the CRISPR at the one-cell stage, and it goes in and edits both copies of the genes that you're interested in modifying, and then goes away, that there's the possibility that what you intend to happen will actually happen. However, there's nothing to prevent that cell from starting to divide before CRISPR finds its target site in the genome and starts editing those two genes. And as a result, if it doesn't edit the two genes at the single cell stage, but rather starts editing at the multicellular stage, then there's the potential that it could edit 0, 1, or 2 copies of the gene in any one of these cells, thus leading to a mixture of cells with basically different genome sequences or genome editing outcomes amongst all of those cells. And if those cells then make up the full individual that is born, then you have what's called a mosaic, where some of the cells have different genome sequences. So there's other issues that arise, but those are some of the major ones. So to just summarize quickly what the claim was in the presentation that was made, and in the news stories and interviews that resulted and associated YouTube videos, I will say I highly encourage all of the videos from that meeting. All of the presentations are available online from the National Academy's website. I would highly encourage you to go watch this presentation, given its historic relevance, and in particular the question and answer afterwards. I think it's just very illuminating about maybe how to do science and how not to do science in medicine. But so the claim from this individual, Dr. Zhanghui He from the Southern University of Science and Technology, was that they had performed CCR5 editing in human embryos. They specifically worked with parents where the father was HIV positive and the mother was HIV negative. The rationale for that, maybe we can get to in the panel, and maybe some of the experts can chime in, but the rationale is dubious, because I would just point out it's important to realize that even if there's an HIV positive father, there are methods available to prevent HIV infection to any children. So this is not something that was necessary to prevent transmission of HIV to the children. He announced that there were 2 girls, twins, born in recent weeks. He showed what he claimed were Sanger sequencing reads from these biopsies from these babies. And what it appears from those sequencing reads is that one of the babies has 2 predicted CCR5 knockout alleles, and thus should be null for CCR5. However, it's not the delta 32 mutation, right? These are other insertions or deletions that would knock out those alleles. The other girl has 1 normal CCR5 allele, and then amazingly and disturbingly, another allele that has a 15 base pair deletion that you would predict would lead to a loss of 5 amino acids, but still expression of a version of the CCR5 protein, of which, to my knowledge, no one has any understanding of what the effect or function of that deletion will be on that CCR5 allele. I will point out that in the Q&A, he was asked, did he know that this was the sequence at the embryonic stage via sequencing of that single cell before he transplanted it? His answer was yes, they did know from that sequencing, and that they asked the parents what to do. The parents said to transplant the embryo, and so they went forward. The sequencing reads indicate that both girls appear to be mosaics, meaning that the editing did happen after the single cell stage, and thus they would be a mixture of cells where these mutations exist and other cells that have normal copies of the CCR5 allele. He also announced that there may be another pregnancy that has established from what they did. So I guess to get the conversation started for the panel, some of the possible ethics and concerns related to what was done here, and this is by no means intended to be a comprehensive list, but I only had so much time to put the slides together. So obviously one issue is transparency. This was something that the world did not know was happening, and obviously one of the things that I think the scientific community called for early on was that any type of new efforts in this area should involve transparency. There's been a lot of discussion around the lack of a medical need and the choice of CCR5 as a target. As I mentioned, there was no reason to think that the children were at risk for HIV infection. And so given all of the number of risks that I described and others that I don't have time to get into, the rationale for choosing CCR5 is unclear, at least to most people. There's a number of issues with kind of the technical competency and accuracy. So I mentioned there's mosaicism, there's a new modified gene expressed by one of these children that we don't know what it does, and that there's, despite some efforts that the scientists made to sequence the genomes of the embryos and the parents and the patient, its current technology is simply not able to exclude the possibility of off-target editing. There's a number of other issues that I'm not really an expert to comment on, but I think we will have some on the panel around the informed consent that was used. He posted online, I think it's since been taken down, what he called an informed consent form that he used with these patients. I think it doesn't look like what a typical informed consent form might look like. And who did the informed consent was another topic. Apparently he individually did the informed consent with these parents. It's not clear exactly what the ethical and regulatory review was for these procedures. And there's also been a number of stories about who knew that this was going on and what could they or should they have done. There's been a number of stories that he had expressed an interest in doing some of this, or that he was planning to do some of this, to a number of different scientists in the field. And so that's another topic potentially for discussion. So this is the last slide. So this is the statement that the organizing committee from this meeting put together on the final day following his presentation. I am not on the organizing committee, so I was not responsible for crafting this. But the kind of interestingly and somewhat controversially, one of the things that they proposed was the need to establish a translational pathway for germline editing. And I think that there's discussion following this about, this kind of gets into what is the translational pathway for human germline editing, skipping over the question of should there be a translational pathway for human germline editing. It basically condemns this report of the clinical use of germline editing, calls it irresponsible, and also recommends an independent assessment. I think it's important to realize that other than the presentation that was given at the meeting with some Sanger sequencing reads and PCR bands, there is no other independent validation that this has happened. And then, of course, they encourage an ongoing international forum for broad public dialogue. But with that, maybe I'll just leave that there and look forward to the panel. Thank you for your time.
So we have, most importantly, 5 seats at the front. Does someone come? 4 seats. So sit down. Right. Oh, sorry. So I don't think it's a case where we think it's Dr. Voldemort. We have not named Dr. He-Chang Kui as the PI of the study, so I'm going to keep referring to him as Dr. He, who conducted the study. I think we should be clear that there is an individual, for whatever purpose he desired, to generate the data, so we shouldn't ignore the fact that there is a PI behind the study. The other thing I want to get away from, so I've spent the last week since we established that this was going to go forward, I read a lot of the comments about the news articles, and it's always interesting to dive deep into the lay public's anonymous postings. So I want to get away from the fact that be objective about the nationality, okay, as well. So I don't want to take the route that it's going to happen in certain countries versus other countries. So I want to get that off the table as well. So we really want to talk about the science and the implication of what's being carried out. To echo Charlie's comments, we have no idea whether this data is real, but I think we have to assume that if it isn't, it can be soon, and I think these are really critical discussions that we're going to have today. So you can clearly see why I referred the Wall Street Journal's question to Charlie, because he is an expert in the field. But if you guys just want to briefly introduce yourself, say why you're here and why you wish you hadn't agreed to answer my
My name is Heidi Cope. I'm a genetic counselor here at Duke. I've been enrolling participants into genetic research studies for the last 15 years. Currently, I work on the Undiagnosed Diseases Network genetic research study with Vandana Shashi.
I'm Vandana Shashi, and I've probably been a clinical geneticist longer than many of you in this room. So I take care of patients in my clinic. I also do research on undiagnosed diseases. And so an important part of my practice and research is to kind of embrace new technologies to help our patients. And this is certainly of great interest to me. I'm not very knowledgeable about the intricacies, but it certainly poses a lot of questions to those of us who practice genetics.
I'm Ginny Fuda. I'm an assistant professor in pediatric and in the MGM department. And my work is on HIV vaccine research, mostly in the setting of mother-to-child transmission.
I'm Isha Ingres from the Initiative for Science and Society, a recovering human geneticist, troublemaker.
So for those of you looking at your watches thinking, oh crap, we've only got 10 minutes of panel discussion, I'm not saying we're going to go until 5:30, but I've booked this room until 5:30, and the panelists have agreed, except Charlie, who has to meet with someone at the back there. So as soon as that guy, don't let whoever it is out of the room, and Charlie will stay. But the other guys have agreed to stay for a bit longer as well. Not until we've exhausted the topic, but certainly until we get a good chunk of the discussion underway. So I'll start first with Ginny. Can you sort of take up where Charlie left off about the requirement for a CCR5 editing, given the fact that germline editing, well, the justification for the approach is that it has to meet an unmet clinical need. So can you speak about whether it was necessary and whether there are alternatives that could have been used in that context, and also the male being affected and the female not being affected?
So for me, it's really difficult to find a scientific justification for this experiment because presumably these were non-HIV affected embryos, and there are other ways to prevent the HIV acquisition. And if the goal was to prevent not acquisition, first of all, their mother is not infected, so they didn't have a risk of acquiring acquisition through mother-to-child transmission. And if it's for long term, one thing to take into account is the fact that the risk of transmission in China is actually pretty low. The prevalence of HIV in China in 2017 was less than 0.1%. So the chances, that these kids, when growing up, will acquire HIV in this setting, first of all, are low. There are other measures to prevent HIV acquisition, and even in the case of HIV acquisition, there are treatments that are available. The fact of using, an HIV-infected father and HIV-uninfected mother is also not really clear, because transmission occurs from the mother to the baby during pregnancy, at the time of delivery, or during breastfeeding, but there is no transmission from the father to the baby. So a possible explanation is that one way to reduce the risk of transmission of HIV to both the mother first and to the baby subsequently, in the case of discordant couple, is the use of sperm washing, so an in vitro fecundation. So maybe it's because as a way for this couple to have a baby would have one possibility for them to have a baby is through in vitro fertilization. Maybe that's part of its justification, but it's not really clear.
Okay. So Dr. Herg collected families from an HIV clinic, as I understand it, ascertained that with the promise of delivering what he quoted was an HIV vaccine, as though ascertained I listened to his talk from a couple of different perspectives in the room at the time, and he was saying that there were 4 people involved, and then when he talked, he said that his postdoc chatted to the parents and discussed the project with them first, and then there were 4 people who ascertained. Heidi, can you speak to the normal process of ascertainment? How it's, and understanding that the laws governing ascertainment vary from one nation to a nation, and a different nation, but how do we do it in the US?
Sure. So in the U.S., there's various ways that we can let patients know that studies are available. If you get the proper IRB approval, you can post advertisements that they can respond to, or a clinician known to them can speak to them about the study. And then if they're interested in participating and agree, then the researcher can speak to them directly to explain the study. And typically, while the PIs are capable of obtaining informed consent, it's not typically the PI who does the consenting process. Typically, it's a study coordinator, a genetic counselor, a nurse, someone like that, who is doing the informed consenting process with the patients. And while we do tend to think of it as a document that is signed at one point in time, it really is more of a process that you are talking to them over a period of time, explaining the research to them, answering all of their questions, and it culminates in the signing of a document, but does continue throughout their participation in the study. And it is typically someone who is trained in speaking to people in a very simple way. So while a lot of the bench scientists are very brilliant scientific minds, they tend to talk in a technical lingo that the average person does not understand. So the genetic counselor, we translate that into a language that the patient can understand and can consent to in an informed way.
So I wanted to go back to the being in the room when it happened, Charlie. Your video is, or the picture around the room of the press interest was pretty informative. And certainly I was watching a video from a guy who was sitting in the front, and you could hear the moderator telling the press that the speaker is not going to move. So you can take one photo, it's still going to be the same photo whether you take it now or whether you go to it. But what was the consensus in the room? Because my feeling was from the questions, as you mentioned, there's a fair bit of anger from people, from not only from the US, but the UK and China as well.
Yeah, I mean, it was it was kind of a weird atmosphere. I mean, there's a lot of media there. I actually talked in the session after him and was very excited to have this kind of a audience, except that after his presentation, everyone left to go write their stories and post their blogs, and there were like 6 people left in my talk. But putting that aside, I think we were baffled, right? Because I think that one of the things so all we knew going into the talk was this report that there might have been genetic babies, and I think that I think for the scientists who can kind of interpret the data, when he put up the data and it became clear that, because it was only after he started showing the data that we realized that these babies are probably mosaics, that there's this CCR5 mutant that should be expressed, but we have no idea what it does. Some of the answers about how he did the informed consent, these were all things that were new and coming out in real time in the presentation. The fact that there might be another pregnancy, the fact that he started with 8 couples, not just this one. So I think that a lot of the dialogue after the presentation was that it was kind of worse than what people had originally thought going into it. And so yeah, a lot of the Q&A I think was from just kind of a baffled perspective.
So I suppose the concern, so I looked at his Cold Spring Harbor presentation in 2017, where by that stage he'd edited literally hundreds of model organism like mouse. He worked in non-human primates, and he'd worked on non-viable human embryos as well. And he was looking at editing and looking at the the off-target effects, looking at indels at that stage. He wasn't looking at mutations. But how can someone who, and in Cold Spring Hub, like granted there's lots of genome editing meetings, but how can someone who's presenting in that sort of forum fly under the radar?
Well, I mean, doing the germline editing in animals is commonplace. We've got core facilities that do this, right? So I think that's most all universities do. So that I think in and of itself is not surprising. And as I had mentioned, there were a number of papers coming out from very reputable places on germline editing for large animal models, and then human embryo editing for non-reproductive purposes. So I think in and of itself, it didn't stand out as an outlier. I think that, in the presentations, to my knowledge, I was there for some of those presentations. I don't remember, and I don't think anyone else has reported it. I didn't think it stood out as a red flag that he was going to try and do this in human embryos that he was going to term. I think that other reports have come out that he had potentially talked to other people offline about this, but I don't think in scientific presentations there was any indication.
Right. And so we've heard about whether it was necessary from Jenny, the fact that they probably haven't been ascertained appropriately from Heidi and yourself about whether it's likely to be done, by someone. I'm not saying the biohacking folks who are modifying frogs apparently in their own garage. This is obviously a lot more serious than that. But Vandana, can you speak to the sort of what's the unmet clinical need? Where do we define the sort of the requirement to proceed with this sort of the germline editing as opposed to the somatic editing that Charlie's involved with his research?
So unmet clinical needs can be defined based on the particular field you're thinking about, the disorder you're thinking about, and what the objectives are. So in typical medicine is if you don't have a specific treatment for a disorder, that's pretty disabling, and existing methods of management just don't alleviate the patient's problems. That's an indication of an unmet medical need. In this case, clearly we've heard that the medical need did not justify. In terms of even germline editing for genetic disorders, at this point it's hard to justify an unmet medical need. If you have, say, a fatal neurodegenerative disorder that's running through a particular family, and obviously it's genetic, there's a way to test for it, you have the option of pre-implantation diagnosis, and you can select embryos that are unaffected. So and that's a much safer way to do it. You don't have all of the unanswered questions you have here. So it's really hard to justify an unmet medical need at this point for germline editing for even serious genetic disorders.
So I know that George being a contrarian at the best of times, so George Church made some interesting comments, and I think his point about making a balanced presentation so it's not piling onto the guy may hold some water, but Misha, what's your opinion when he says, well, as long as the kids are okay, it was an issue of paperwork and someone was going to do it at some point, so it should be okay?
Well, so I spent several years following George Church around and wrote a book about him. I have a great deal of affection for him, and my fondest hope is...
Did you just get a plug-in for a book?
It's out of print anyway. My fondest hope is that he was somehow taken out of context or he didn't mean it. I haven't spoken to him about it. But, that's a very consequentialist view of the world, that, if the babies are okay, then it's all good. I think we've already heard about problems with the informed consent. And, you can still find the informed consent online, and I would encourage people to read it. It does have some things that reminded me of my 5 years on the Duke IRB. A lot of, if things go wrong, it's not our fault kind of language. And there's also a fair amount of language about, we reserve the right to use your likeness. If we disclose any secrets, you're not allowed to talk about it. And I think it sort of speaks to one of the larger problems that a number of people have commented on, which is, this guy co-opted the Associated Press, who knew that this story was coming, and went on YouTube. So when I go on Twitter and I see people comparing him to Edward Jenner, it really makes me uncomfortable. Edward Jenner did not go on YouTube.
So what do you think about if it wasn't him, it would be someone else? And I think Dr. Hu actually said that in one of the interviews I watched. He finished with, if it wasn't me, it would be someone else. I don't think it's like the Wright brothers. The Wright brothers didn't fly. You've got a bunch of French guys who will be first in flight. But I think it's if it wasn't going to be him, I think it would be, given the the accessibility of the technology that Charlie talked about.
Well, that may very well be true, but you would hope that someone else would have done it better, more ethically, more carefully. And so again, to go back to Edward Jenner, yes, you could say he risked the lives of the children he inoculated with smallpox or cowpox, but he also actually collected data, and there were people in Turkey who were doing inoculation, and I think it doesn't have to be done in sort of cowboy fashion right out of the box. And I think a lot of the anger that we see on the video that was expressed at that meeting is because that approach now redounds to everyone else in the field and really jeopardizes the field.
So the point that you raised before, and this is open to Ginny and Vandana, about in the consent form it says that they will follow the twins until they're 18. So how do you, as physician scientists or even physicians, how would you, and it's a very difficult question, how would you counsel kids who they'll probably be told or they will find out somehow? How do you counsel them when they're coming into the clinic about their HIV risk? And we haven't talked at all about the fact that CCR5 knockout makes the girl who is homozygous more susceptible to other types of viral infection. So how would you talk to them about that sort of instance?
I can add to this.
Yep.
Of course, this is such a unique situation. I don't have a situation I can compare it to, but the closest I could come to is when we test kids asymptomatically for something. And the only justification to test a child for something when they're not symptomatic or something is when they're at really high risk for something and there's an intervention for it in childhood. Otherwise, we always recommend waiting till they're much older before we talk about risks and testing. So these children are in a situation where they already will know their status and we don't know the benefits. It makes for an extremely hard situation, even in clinics when there's a particular indication, there's an actual intervention that's necessary for them to optimize their health. These are difficult conversations at best, and I think this makes it so much more complicated.
Yeah, and I would add that in research studies, when you enroll, you are not signing a contract. This consent form actually said it was a contract, and you, as a participant, have the right to stop participating at any time. So these children cannot consent for themselves. At some age they will be able to, but they can't currently. So for fetuses and babies and young children, parents make consent decisions on their behalf because parents are thought to have the best interest of that child at heart. So while the consent form said that they would follow these children for 18 years, the parents should have the option to stop participating at any time if they don't want their child to come in for the next 18 years. Also, the things he clearly in the consent form spelled out the types of testing you would want to do on those children over the course of those 18 years. And some of the words, it said surgery at one point, and testing for gonorrhea, which these things seem completely irrelevant to the purpose of the study.
Right,
So I think the consent form does say you can withdraw, but it also says that if you withdraw, I think after implantation, there's a pretty substantial fine.
So Charlie, can you, and it's difficult to put yourself in the minds of parents who believe they're doing this for the future safety of their children, but can you understand at all from Dr. Hu's perspective about continuing on with the embryo that was heterozygous for the knockout? Would there be any scientific rationale for doing it?
Yeah, I mean, taking a step to the earlier part of your question, I mean, I wouldn't want to put myself in his shoes in this scenario, but to relay some of the things that he said in the Q&A
Right.
Afterwards, and it gets to some of the earlier points. You know, he was asked a lot of these questions, which is why I encourage everyone to go and watch it themselves. And he was asked quite a bit about, you know, how can this possibly help these children or protect them?
Right.
And he gave a number of answers, none of which seemed to answer the question about those individuals, but more were about society more broadly speaking. So he referred to the fact that he's been to villages in China where the percent of HIV positive individuals is 30%. And how that was, you know, something that drove him to do this. He brought up the fact that the father of these two children had expressed to him no interest in continuing to live until now, or something along those lines, until now these two children were born. Now he had new meaning in life and was going to work hard and provide for his family. And so he was using that as an argument of how this was good. The issue with the 5 amino acid deletion, that one is harder to wrap, even more harder to wrap your head around. He was asked the question, and his answer was that he asked the parents, and the parents told him to implant it.
Right.
Which is, I think, I guess I'd be interested in a genetic counselor's perspective on that move, but that was surprising to the audience.
Generally, in genetic counseling, we do like the patients and parents to be able to make decisions, but since this is within a research setting, that would have been a discussion that you have at the outset, that if the created embryos did not end up with the genetic change that you were aiming for, that those would not be eligible for implantation. So that wouldn't have been an option to implant at that point. But we do like to have as much autonomy and self-decision making as possible.
Right. So Ginny, can you speak about the susceptibility to other viruses with the CCR5 knockout?
Yes, and the first thing that I first want to point out is that even individuals who are homozygous for the Delta 32 mutation are not completely resistant to HIV, because it's not CCR5 is not the only receptor that HIV can use to infect cells. And there have been some cases of individuals who are homozygous who actually acquire HIV infection. They are far more resistant than the rest of the individuals, but they are not completely protected. And it is also known that these individuals are more susceptible to some other viral infection, notably infection with flaviviruses. They are more susceptible to the West Nile virus, to some sort of encephalitis. There are some reports, even though they are not very consistent, that they may be more susceptible to hepatitis C. And there are also some studies suggest that they could be more susceptible to some other immune disorders, such as multiple sclerosis or lipid-associated nephritis. So it's not completely, innocuous to have this protection.
So he did say that he was going to use sort of in vitro testing using T cells to determine whether the HIV resistance would work with the knockout? Is that like a standard means by which you can determine resistance to the virus?
So yes, you can do in vitro experiment and try to see the susceptibility of the cells to infection by HIV. But an important point, that have to be kept in mind is that we don't really know what is the phenotype of the T cell. We know that they don't have the same, they are heterozygous, at least one of them, and they all have the mosaic phenotype. So what exactly the T cell express? Are the cell expressing two different receptors? Are there some cells that are expressing the mutated receptor and some that are expressing the normal receptor? What is the distribution of the cell? Like, are the cells circulating in the blood the same that the ones that are at the mucosal site where the infection occurs? All of that is not clear. So I'm not, it can be informative, but it will not really give a definitive.
Right. Okay, so I'm going to ask Misha a couple of curly questions, since I haven't checked any off on my list, and then we'll open it up for questions to the audience. In my reading of the public commentary about the findings, one of the questions I thought was sort of an interesting one: is this a problem about the technology and not morality?
No.
Do you want to elaborate?
I mean, if you look at sort of, if you've read Ed Young's piece in the Atlantic.
Yeah, I'd recommend you read that from December 15th.
Which
December the 15th.
enumerates 15 things, 15 problematic things with this experiment. They're not necessarily unique to germline editing of CCR5. It's an investigator who sought counsel, got counsel, ignored that counsel. We've seen this in other instances through history. The science, I think Charlie illustrated it beautifully, and there was another piece on STAT that went up either today or yesterday, further diving into the weeds about how, which alleles were actually present and the extent of the mosaicism. And so even by its own, by the PI's own standard, this failed. And we've talked about the consent form. The consent form, I would say, is disingenuous at best. And all of this was done in secret. So again, David Baltimore and Jennifer Doudna and other people at that meeting have lamented repeatedly, that this was a failure of self-regulation and it was a lack of transparency. And those things are not just things that ethicists wring their hands about, but they are counter to the norms of science. Science is supposed to be self-correcting and falsifiable, but if we do it in a dark room, then that's just not possible.
If humans are taught that we control our own destiny, why isn't this just seen as humans controlling our own evolution?
Well, that's a bigger, longer-term question.
Right.
I mean, the near-term thing is, Charlie showed the New England Journal paper where using, zinc finger, you can do this, but Dr. Hood did not do this. And so if that's what controlling our there's the question of, is this aspiration or ambition something we should pursue? And I don't know. I mean, in my view, that's a secondary or tertiary problem, given all of the very long laundry list of things that went wrong here.
So, open it up to you guys. Crystal, do you have any questions?
I've submitted a lot of questions, and I also was in a dinner with Charlie yesterday, so I asked a lot of questions. So I'll let
All right, good. Okay, yes, please.
So my question is for Charlie. So obviously there was a lot of, you mentioned there was a lot of surprise from the scientists during the presentation. For the rest of the conference, was that basically all anybody could talk about? And were they focused on the science, the morality, or was it a mix?
So I mean, the presentation is a 3-day meeting or 2.5-day meeting, and the presentation was kind of in the middle of the second day. So yeah, there's a lot of speculation leading up to it. The media presence kind of made it hard to ignore. This was the only. It wasn't really a scientific meeting, right? This was kind of the only new science. There might have been some other new science, but it was more a meeting about discussion and not new science. This was maybe the only new thing that was presented. So yeah, it definitely drove much of the discussion. I do think, there was a lot of discussion about the morality and ethics of this. This was a meeting where probably there were more experts on those topics in the room than there were scientists by design, because that was the point of the meeting. So there's a lot of discussion about it, and similar to what we've talked about today and what's been written.
Can any of you speculate on how this might affect regulation in the U.S. on genome editing?
So, in fact, the reason I'm repeating it isn't for your sake, it's for the stream. So, affecting regulation in the US.
So germline editing is effectively illegal, and my colleagues will correct me if I'm saying something wrong, but my understanding is, the FDA views modified embryo as a drug, as an unapproved drug, and it is verboten. Now, how that's enforced, I don't know.
the NIH also will not fund any studies that involves this kind of germ editing or research along these lines. Again, as Misha said, we don't know. There isn't a specific oversight of this.
Sorry. I said the NIH does not fund these kinds of studies for germline editing, so that's another way of how the government does not approve this, of course it comes from DHS. And also the FDA, I don't think has any one specific section or protocol that oversees this. As Misha said, they look at it as a drug. And I think this will probably spur some more discussions around policy for this. There are countries like UK that will allow this for a certain number of, up to a 14-day age embryo and things like that. That is not allowed in the US. But I think it will spark some discussion as to if this is so important. And I think this research is so important. How do we develop policy around that? And I think it will certainly stimulate that kind of scientific discussion in this country.
Israel? Oh, sorry.
Well, I was just going to add, I think my understanding is that there's a law passed by Congress that the FDA is not allowed to even look at or consider any proposals that involve edited human embryos. So they're not allowed to even look at that. I think one of the concerns is, will this have any effect on regulation of the somatic cell gene editing therapies, like, I listed a number that are moving into clinical trials and are very promising for very horrible human diseases. And I think that the hope in this community is that this won't have a negative impact on that. And Francis Collins was quoted in his story about thinking that would be very unfortunate if this impacted the promise of those therapies.
All right.
I do worry though about the impact on patients being willing to participate in genetic research studies. I can say that after Dolly the sheep was cloned, that That was my most common question from participants enrolling in research studies is, are you going to clone me? And probably a large reason why people chose not to participate in genetic research studies. So I do think that this kind of research can scare people away from participating in valid studies.
Isabel?
So though I agree, like the country in which this was done is pretty much irrelevant for this discussion, but I do, from some of the things I've read recently, there's controversy about, in different countries, there are different guidelines for what is legal and what is not legal in terms of research. Do you think that this will cause the formation of a group analogous to something like the United Nations that will oversee research on a global standpoint, and do you think that is necessary with how we're moving with genetic technologies?
So my understanding is that it's illegal in China, and I also understand as of yesterday late that Dr. He is under house arrest. So he is escorted from the conference, and I think that sort of speaks to the level of consternation from the Chinese government. But I think it's a really interesting question whether there's UN-type oversight for this sort of research.
Within the last couple of days, the World Health Organization has come out and said they're going to take up this issue.
Yep, down here.
Yeah, so it was said before that at the, like in the room at the summit, that there were more bioethicists and people that kind of think about science in that way than scientists. Do you think there's a need or space to start giving scientists frameworks to start thinking about their work in a way that is looking at like society and able to tackle some of these ethical considerations as we're conducting our research?
Yeah, I think there's probably a good need for that, right? Both in during the education process, when you get your PhD and postdoctoral training, and afterwards. I think that I mentioned lots of these genome editing meetings, and lots of them have ethics sessions, even at the science meetings, right? And I think that hopefully something like that with some more education around those issues will help prevent someone who's, in this case, trained as a biophysicist on DNA sequencing technologies from moving into this area. And I think maybe that's part of the issue is that one of the concerns with something like CRISPR is that it's fantastic that it's so easy and robust and cheap and fast, but it takes something where you can have serious medical intervention, it puts it in the hands of anybody, so maybe you need more people who have the knowledge to how to implement something like that, have more of the background.
Yep, over here.
So the Delta 15 allele has my curiosity going. We had an unknown.
Can you just speak up a little?
I'm sorry. The Delta 15 allele has an unknown phenotype on these T cells. And these two girls are very real human beings who will go on to lead lives. Did he unintentionally create a genetic disease with these mutations that may propagate? And is it inevitable that that might happen with other trial agents?
Notice that I'm looking at Charlie, I'm sort of got the eyes down.
Yeah, so the question is, did the 15 base pair deletion create a genetic disease? I mean, I think the answer is we don't know anything yet, right, about what that created. Right. As was mentioned, CCR5 does all types of things and probably does a lot of things that we don't know about in lots of different cell types. And that's why, one of the arguments for the original pursuit of CCR5 knockout in T cells was that you're only modifying those T cells, only the cells that you took out and put back in, and not every cell throughout the body. And you're doing it where there's a medical need because it's an HIV-infected individual. Obviously, in this case, it's different. I think that whether it's a disease, we'll have to wait and see, what type of phenotype might manifest, and I'm sure that this is going to spur people to clone the 15 base pair mutation and test for its function. One of the things that was discussed at the meeting was, what does it mean for these people in terms of these two girls for their progeny, right, and their decisions to reproduce, and even between the two of them, given that they're now twins, but designed differently at the genetic level, right? So I think that there were questions about that, and there weren't necessarily great answers, but yeah, these are real individuals who are now going to pass these things on.
I just wanted to stipulate that we don't actually know for sure that these two girls are real individuals. There's been no proof. With human cloning, there were 3 separate publication saying that humans had been cloned, which then all turned out to be false.
I wanted to add a comment to your question about ethical training, so these things can be prevented. At least in this country, in most other countries, there are IRBs and ethical boards and human subjects requirements that potentially would serve as checkpoints.
That said, Mark Yarborough had a great piece in Stat the other day talking about how this person did their PhD at Rice and a postdoc at Stanford. So again, another reason not to throw stones at China. And again, consulted the Hurlbut's father and son bioethicists and sought counsel from many people. So, you know, I think that's occasion for us who teach bioethics and responsible conduct of research to think about how we can do what we do better.
But I think my understanding was that they said don't do it, and he went ahead and did it anyway. So I think it's like the British policeman used to be, who walk around with a truncheon who would say stop, or say stop again. It's a bit like that. Do we have the international law? I have no idea whether they're going to make an example of Dr. He. So I think there has to be consensus, and Charlie's listed it and appropriately left it as the last slide, that we need transparency. We need some sort of international governance, but it's how to control the rogue individual is going to be a challenge.
And it's clear he as an individual broke all the laws, right? It wasn't that the systems let him do that. He hid it from everybody else, so it could happen anywhere.
Because I think there's questions now about the signatures on the review of the consent being forged. The hospital who supposedly allowed him to proceed said they hadn't seen the form before. So there's sort of multiple levels in which he's managed to get through. We'll have two more questions, and then you all have homes to go to or pubs to go to. We have homes and children. Yep, over there. Yep. Yes, you.
Yeah, okay. So, as Charlie brought up, they say at the summit that they recommend an independent assessment, and Heidi also clarified that we actually don't know. Did they decide who is going to independently assess what actually happened, and if the mutation are actually present, and how will the scientific community be kept in the loop?
I think the university that Dr. He has his principal or primary appointment with has already instituted an independent review of his work, and he has been on paid leave since February, I think we mentioned, where he's sort of preparing for the coincidental release 2 days before this conference. So I think that's already well underway, and I would have thought they'd be going over all of his data.
One of the interesting issues that came up is that apparently it is against the law. There's a law in China that you cannot reveal the identity of an HIV positive individual. So that also plays into this whole idea of who identifies the family, or figuring out who the family is, etc.
Okay, one last question over here. Yep.
Just a follow-up to your response there. Just to make sure I understand this properly. The university that allowed this to occur.
They didn't know that it was occurring, so they didn't allow it. Right,
But it happened under their nose, and now they're doing the independent review?
No, there's an external independent review.
Okay.
Yeah. All right. So since that's not a question, well, it's a question. All right, right at the back. Yep.
So I feel like I can ask this question because I feel like there are lots of Chinese students from China come here
Yep.
in this room. No one really ask questions, and the Chinese student, I guess I can kind of have partial response to the previous question and my own question. So the previous question, I read lots of Chinese report of this. So the person, so it was claimed like this. The person walked in the public university before, but when he tried to carry on this research,
I think, from my perspective, I think there has to be some sort of unification of standards. I think that, we've spoken several times about transparency, but I think the unification of what's acceptable, the unmet clinical need, the way that patients are ascertained, one of the things is the sort of broad consensus from the public. I think we'd never get that, honestly, after reading the comments. So I think as young scientists and made light of the fact that you're all going to the pub, you actually should all be back to the lab. I think it's beholden upon you guys to make sure that you hold yourselves responsible to it, or to a certain level, accountable to a certain level. You hold your peers accountable to a certain level, and that sort of pervades the level of science. That we take into account things like ethics, we take into account consideration of the families, the patients, the type of rigor that we have for our science to make sure that when we say something doesn't have an off-target effect, we actually know that, we're not claiming it. And those sort of things, I think, really starts with you guys. And I think by internationalizing science, where everyone is speaking with the same voice instead of pointing fingers at one nation, pointing fingers at one lab, I think they're really, really critical going forward. So it really does start with you guys.
What was to say about the self-regulation point? There was a lot of debate. I think maybe it's sometimes heated at this meeting about, is this an example of where self-regulation failed or not? Some were making the case that clearly this was a case where self-regulation failed because this happened. But others would argue, contrary to that self-regulation, the community said that you shouldn't do this, it was against the law, and it happened anyway. So whether or not one individual doing something against the law in secret qualifies as a failure of self-regulation or not, I think is open to interpretation.
All right, so we'll end there. So thank you very much for your interest. Many thanks to Charlie, again, for suckering me into this. And